Hello, botanists. It's me, Stephanie. You'll be seeing my face again and hearing my voice throughout today's virtual lab, where I'll be talking about root development. And so, before you get started with that, I want you to think about the biggest tree that you've ever seen in your life. Maybe it was a redwood up here in Humboldt, or maybe it was a giant sequoia, which are even bigger in volume than redwoods. Or maybe it was a huge Douglas fir or an oak tree or something like that. And imagine that huge tree that you can see above what you can see above ground, it has a similar biomass below ground in its roots. It must be absolutely massive underground, and we can't really see that, but we're going to talk about how roots develop into these large woody structures. I also want you to imagine that largest tree that you've ever seen, and imagine it as a very young seedling. At one point, it must have been very small and young. And imagine that very, very first primary root that first emerged from the seed. And that's where we're going to start talking. Enjoy. All roots go through primary growth when they germinate from the seed. Primary growth results in an increase in length. Some plants go through secondary growth, and this is when a root actually increases in width or girth instead of length. So in this picture, we've got a seedling, and notice it's got these root hairs all over it. Root hairs are the only place on a root system where absorption is actually taking place. So here is an illustration, a diagram of a root. If we were to take a slice using a very thin razor, right down where I've drawn these lines here, and slice it out of there and then sort of turn it on its side, that's what we would see. We would see this image right here. We can see a lot more in this photograph that was taken through a microscope than we're going to be able to see in the diagrams that we'll be drawing for the rest of today's virtual lab. So it's really important to kind of ground ourselves in this image. What we can see very clearly are individual cells. And at the very base, sort of towards the lightest blue part of the very tip of the root, that's just called the root cap. There's actually no cell division. There's not much cell division going on there at all. That's just sort of a protective barrier for the tip. There is a lot of cell division going on immediately above that light blue tip, where most of the cells are in this sort of darker region. And from that darker region, there are some kind of different colors radiating up and out. So on the very outside, there's a pinker region, and that is where epidermis is going to be born from the primary meristem protoderm, which I'll have labeled in just a sec. And then those really obvious dark and then light lines down the middle, those lines represent the division between the ground meristem, or the ground tissue, and the procambium, which is in the very center of this. So again, most importantly about this photo, what you're seeing is the very small cells. And when they're small, they must be very young because they just divided via mitosis. You see those only at the base, and that is the root apical meristem. Above, the cells get much bigger, and you can sort of see them with your naked eye in this image. The cells have elongated above that root apical meristem. And that's why they're more visible to us. They are getting older, and so they're bigger. Moving on. So in bright cyan blue, we've got the RAM, the root apical meristem. Now, something I've accidentally left out of these diagrams are the root hairs. The root hairs would be attached on the outside of that light yellow outline, which is about to be labeled. So the very base, sort of protecting the root apical meristem, is the root cap. The root apical meristem, again, meristem, this is just a word that means where lots of cell division is happening. And so it's going to be important for the plant to protect that area, and the root cap does that, so that the first thing in contact with the soil is not the root apical meristem. It is this cluster of of cells called the root cap. So the very outside layer you can barely see, labeled in or outlined in yellow, is the protoderm. This is the meristematic primary meristem that will give rise to the epidermis. The green layer inside of that is ground tissue. And then the purple, that inner part of this root longitudinal section, is the procambium. Okay, we're going to use this page throughout this virtual lab. We're going to trace the development of different tissues all the way to a very old root. So the root apical meristem is where all cells are born in the root system. Whether we're talking about primary or secondary cells, the original cell is born in that root apical meristem. The root apical meristem then gives rise to those three primary meristems, which we've talked about: the protoderm, the ground tissue, and the procambium. Okay, so we are watching this root grow and develop. And now the tip of that root is not literally staying in the same place as it's shown in this picture, with it's kind of showing like it almost looks like cells are being added to the top of this. They're not. The whole root would be going down into the ground. And now we've kind of zoomed in here. We've got some new colors. In the very, very center, we have this pink, and notice that that's not the first thing that's developed. If the youngest, again, this is the growing tip closer to the root apical meristem. Out of all of our new colors that were just added, it appears as though that light blue or maybe that bright green were added first. And this means they developed first. Well, the light blue is phloem here. This is primary phloem. And notice how that light blue, it's coming right out of the light purple, which was our procambium. So this is a sign that primary phloem is derived from our procambium. It makes sense that the phloem would develop before the xylem, because remember, phloem is bringing sugars and carbohydrates from the top of the plant to the bottom of the plant, or from the leaves, in theory, where those sugars were made, to the root apical meristems, where sugars are needed with lots of cell division, lots of mitosis occurring. Sugars and carbohydrates are the energy that's going, or are the source of energy to the cells that are dividing. Okay, so we've also got a couple of other things being labeled here. Once the phloem has developed, then it would make sense for the primary xylem to develop. This is where the water that's being absorbed from the root hairs is going to first make it into the center of the root, which will then go up towards the leaves, where the leaves need water. All right, the pericycle is the layer that's in bright green here, just outside of the xylem and the phloem, and it is also derived from procambium. Notice that our ground tissue, the light green, has gotten darker green, and it is now considered cortex. The layer just inside of that dark green is the endodermis. This is where we've got a Casparian strip of waxy cells that will prevent water escape. back to this page where I told you we were going to continue building. Now, from our 3 original primary meristems, we've got primary tissues. Protoderm is sort of the simplest because it's just producing mature epidermis. The ground tissue is producing 2 different tissues, though. It's got the cortex, which is usually parenchyma cells. This is kind of the majority of what you're going to see in a root, usually. And then just to the inside of the cortex is the endodermis. Now, the procambium is a bit more complicated because it's producing a few different types of cells, and it's actually also not producing a couple of cells. In other words, some of that procambium is just going to remain as undifferentiated procambium for the time being until it gets a little bit older. The other tissues that we see derived from the procambium are the primary xylem, the primary phloem, and the pericycle. Here, the cortex is labeled by a red arrow and the vascular cylinder labeled by the orange. This is a cross section through a root, and this is the longitudinal section of our diagram with the colors coordinated. Here, the red arrow is cortex and the orange is the vascular cylinder. There's variation on the relative sizes of vascular cylinder to cortex based on species. Now we've blown up the vascular cylinder from the picture on the left to show you the closer up version of the vascular cylinder of the picture on the right. We've circled the primary xylem in pink. But when we look at a longitudinal section through a root, we do not see the X or the star shape pattern formed by the primary xylem. You can only see that in a cross section. The reason for that is, imagine drawing an X on a sheet of paper or something, and then turning that sheet of paper on its side. Well, all you're going to see is the side of that sheet of paper, and you won't see that sheet of paper was in the shape of an X. You will just see a line, and that's what I'm sketching in the bottom right here, is essentially just a section of a root showing what that X of primary xylem will look like relative to its phloem, which will be on just the outskirts of what appears to be a line of primary xylem, shown here in pink. The primary phloem is shown in light blue, and then the undifferentiated procambium is shown in light purple and labeled UP. In bright green, we have the pericycle. Up until this point in the lab, we've been talking about primary growth, primary development of a root. We've talked about the mature primary tissues, including the xylem and the phloem, and the cortex, epidermis. And for the rest of this virtual lab, we're going to talk about not how a root gains length, but how a root gains width. This is called secondary growth. And it has a very exciting beginning, which you will see now in this following animation. Root development. This video will review root anatomy and the origins of secondary growth. Please pause the video as necessary to take notes and test your understanding. The image on the left is a model of a young root seen in cross-section. Let's review. The epidermis. This is the outermost layer of cells. It was a product of the protoderm during primary growth. The layer inside of the epidermis is the cortex, the ground tissue of the root, which is filled with large, thin-walled cells. The cortex was the product of ground meristem. The vascular cylinder on the interior of the root, this is the central portion of the root. This was the product of the procambium and will be the main focus of this video. We'll now explore the 4 parts of the vascular cylinder. Starting with the inside, highlighted in red, this is the primary xylem. These are the vessel elements and tracheids that are responsible for conducting water and minerals along the root. The primary xylem is often lobed and can have 2 or more ridges. This example has 4 ridges. Now in light blue, these are the cells that conduct carbohydrates produced in the leaves all the way down into the roots. Notice how thin the walls of these cells are compared to the cells of the xylem. The primary phloem is located between the ridges of the primary xylem. Now highlighted in a little darker blue, there are 1 or more layers of non-vascular cells surrounding the primary xylem and phloem. In young roots, these cells can remain meristematic and can divide to produce more pericycle, lateral roots, or the vascular cambium, responsible for secondary growth. The endodermis is a single layer of cells that is technically the innermost layer of cortex. The cells of the endodermis are characterized by the presence of a Casparian strip, a band-like region of cell wall containing a waterproof suberin compound. The Casparian strip ensures that all the water flowing from the root to the rest of the plant must pass through the protoplast of the endodermal cells. The vascular cylinder. This is an actual root cross-section that has been stained to make the difference between cell types more obvious. Please pause the video and see if you can identify these 5 tissues. Secondary growth. Now we will explore secondary growth. This is growth that is not initiated by the primary meristem, but by the vascular cambium. The vascular cambium arises from meristematic cells located between the primary xylem and phloem and the portion of the pericycle opposite the primary xylem poles. These cells are highlighted in yellow. Secondary growth increases the girth of stems and roots and is responsible for the production of wood and bark. These cells divide and they grow. And the innermost daughter cells differentiate into xylem. This is called secondary xylem, as it was derived from vascular cambium. This process of cellular division and differentiation continues, producing several files of secondary xylem cells. Now the vascular cambium is a continuous ring, 1 cell layer thick. After nuclear division, the cell plates begin to form. The cell wall is now complete and 2 daughter cells are formed. Then the daughter cells elongate. And the inner files of cells differentiates into more secondary xylem. To produce secondary phloem, the vascular cambium divides as before, but this time it is the outermost file of cells of the vascular cambium that differentiates. It is now sandwiched in between the 2 types of vascular tissue and continues to produce secondary xylem to the inside and secondary phloem to the outside. All right, let's take that new information and add it to our diagram, our flowchart of development. The vascular cambium that we just learned about is derived from 2 different types of cells. It's derived from pericycle and from undifferentiated procambium. The vascular cambium is then the new meristematic tissue. It will continue dividing and expanding the circle around as it's getting wider. And it's getting wider because it's adding secondary xylem to the inside of that vascular cambium line. It's also adding secondary phloem to the outside of that vascular cambium line of meristematic cells. All right, we still have primary xylem in pink. We now have secondary xylem in red. The vascular cambium is shown here in black. I know that because it's just outside of the secondary xylem. Just outside of the vascular cambium, we have secondary phloem in dark blue. There's still some primary phloem outside of that secondary phloem. The pericycle is outside of the primary phloem still, and then we have the endodermis in dark purple. Now I've got continuing cortex and epidermis in green and yellow. Okay, so our plant is growing up a bit. Here in that bright blue, we still have that representing the root apical meristem. But in this older plant on the right, it's produced a couple of lateral branches, and so now there's 3 root apical meristems, all sort of shooting out in whatever direction in search of water and mineral nutrients. And notice how there's root hairs associated with the tips of these stems, of these roots, but not so much of the older parts of the roots. So let's say that there was a cell that was born right in that X. It does not travel down with the tip of the root. No, that does not happen. It does not end up at a lower part of the root in the future. No, the cell that is born, say in that young root, right where that small X is, it will remain in that exact literal location in space. The root will continue growing down, but cells are built on the outside of it. Take a closer look at a cross section through a lateral root. So when we look at a lateral root, a young lateral root, it looks exactly like the very original primary root that we were first looking at. And that's because it literally is. It's just another rendition of the same thing. But this time the lateral root and the root apical meristem that is forming that lateral root, they form from the pericycle. Remember that inside of the vascular, inside of the vascular cylinder. That is where lateral roots are born, from pericycle. So what happens in reality underground is that these lateral roots branch and they branch and they branch again, and you end up with these dense networks of very, very tiny roots that have seemingly bajillions of root tips. Well, remember, the root really needs root tips because that's where the root hairs are and that's where absorption is taking place. I want you to remember that a cell does not it's not born and then it does not travel down the tip of the stem. No. Instead, what's happening here on the picture on the left is we have a whole lot of root apical meristems. It's at all of those tiny microscopic root branch tips. There's more than I have dots available. Is where the root apical meristems are on each of the lateral roots here. Okay, so we've been talking about the vascular cambium. That's what I've drawn here in black first. Now, everything that I'm filling in dark red to the inside of the vascular cambium, we know what that must be. It's secondary xylem, except for that one little tiny remnant strand of primary xylem in the middle in pink. But all of that secondary xylem produced from the vascular cambium is what we call wood. It truly is just secondary xylem. Now, on the outside of the vascular cambium, we're getting more secondary phloem. As time goes on, we're just getting more and more secondary phloem because that's always going to be produced to the outside of the vascular cambium. And since there's so much secondary xylem being built up inside, it's really pushing everything else out, including those light blue primary phloem strands. Those are all getting pushed out. The pericycle also is getting pushed out. Remember, the pericycle is still around, and actually it's got meristematic cells inside of it, and that's what we're seeing. What happens to the pericycle is, as it matures, it starts to produce something called cork cambium. Yes, we have a second type of secondary growth, and we know it's secondary growth because it's from a cambium layer. So vascular cambium, that was the first type of secondary growth we talked about. That's producing vascular tissue. The cork cambium is producing essentially bark and bark products. And so that's what we're going to kind of zoom in on here. While the secondary xylem and all of the cork cambium and its products are being produced, it's literally pushing out the cortex and epidermis, those results of the primary meristems and primary tissues. It's no longer sort of needed. That cortex, including the endodermis and the epidermis, it's just shed off over time. The new outer layer is going to be the cork cambium, again in bright green here, and its products. Totally just shed off. No longer needed. And that's happening as the cork cambium is expanding. And the cork cambium and its products together are called periderms. When there are multiple layers of the cork cambium and its products, they're called multiple periderms. Okay. Just remember the epidermis was in yellow. The endodermis was in the dark purple, and the periderms are what we are going to talk about now. I've already told you the periderms have to do with the cork cambium. The cork cambium is derived from the pericycle, which we have here in bright green. The periderms is that whole outer layer of the tan and the green and the brown. The green line, being the cork cambium, is dividing. It's this meristematic tissue again, since it's a cambium. It is dividing over and over and over again to the inside and to the outside. And pericycle cells, meristematic pericycle cells, are also sort of remaining in the area for potential future use. So that cork cambium, as it's dividing, it's producing some cells to the inside, which are called phelloderm, and it's producing cells to the outside, which are just called cork. So that's what I have here. Cork is produced to the outside. Actually, sometimes you hear the word phellum instead of cork. Cork and phellum mean the same thing, and actually cork cambium is sometimes called phellogen. Cork cambium is phellogen. But phelloderm is always the product of the meristematic cells that are dividing and differentiating to the inside of the cork cambium layer. And then just to finish off all of the other labels here. The secondary phloem, the primary phloem, remnants, which will be scarce by this time, maybe by the time it's about a year old or so. Maybe at the top of this image, about a year old. And then again, that darkest red is secondary xylem, which is also known as wood. By wood, I'm specifically talking about the material of secondary growth that is inside of the bark. Which begs the question then, what exactly do I mean by bark? Okay. Well, bark is very specific, actually, when we look at it in botanical terms. The outer bark is the periderm. The inner bark is the secondary phloem, and together, the secondary phloem plus the periderm layers are bark. Yes, all of those layers combined are bark. Secondary phloem and the periderm, including the cork cambium and the cork and the phloderm. Now, secondary phloem alone is considered inner bark. The periderms are considered outer bark. So this is a real image of a microscopic look through a young root. And we can see here in pink, I've just circled a star of primary xylem that we can still sort of pick out here. That means that everything outside of that before the vascular cambium must be wood. This is our first layer of wood. This is our secondary xylem. That's sort of in that very, very light red pen right now. The vascular cambium is visible as a very, very thin, sort of pancake thin layer of cells to the outside of that secondary xylem. You know it's the vascular cambium because it's the thinnest cells. The cells to the outside will be expanding because they're becoming phloem, and the cells to the inside will be expanding because they're becoming xylem. And so I'm kind of circling it here. I'm labeling it here in this thin black pen. That is the vascular cambium. And that means that everything to the outside of the vascular cambium will be bark. First, we've got our secondary phloem, which I'm labeling here in this blue bracket. There are secondary phloem fibers mixed in. That's why there's sort of a bunch of different colors of cells and different looks of cells. We've got sieve tubes and their companion cells. We've also got fibers there. Just outside of the secondary phloem is where we'll first see the cork cambium. The cork cambium, I'm sort of dotting here in a bright green. It can also be sort of hard to find, but it's going to be the thinnest layer of cells. Again, since it's a cambium layer, just like the vascular cambium. It's going to be dividing over and over again, and so you would expect the cells to be the smallest there. They are the youngest. They were just born via division. Via mitosis. So everything in between the secondary phloem and the cork cambium will be phloderm. Everything to the outside of the cork cambium, the very outer layers, is just straight up true cork. Wouldn't really be happening like this. The cells will be growing just at the tips. Here, the little blue dots are the root apical meristems. Remember, there will be tons and tons. This is just a diagram, even more. And you know what there will be even more of than root apical meristems? Root hairs. There will be so many root hairs all over this. Imagine how much water is needed for the tree up above ground. There's got to be a ton of root hairs, a ton of surface area in order to account for all of that water that is needed. Now imagine we take a chunk of the older part of the root. Okay, so again, we've just zoomed in here on a chunk of a longitudinal section through an older part of the root. In the black was the vascular cambium, labeled VC. Everything in dark red to the inside of the vascular cambium is wood, secondary xylem. And these lines here, these longitudinal lines represent year growth increments. So in the very center of this longitudinal section will be the youngest part of the stem. And then as you go out, you'll count the year increments until you get to some number of however old this tree is. Now, just outside of the vascular cambium is that secondary phloem, and then what I'm drawing here are those different layers of periderms. Now, the cork cambium, it's not just a single layer. We've got multiple layers sort of on top of one another, and part of the reason for this is as the secondary xylem is expanding, it's really kind of making the whole cylinder, if you will, of root wider, and so it kind of cracks on the outside, and it needs to continuously heal itself with that cork cambium and the cork and phyloderm. These concentric rings that I'm highlighting in red represent a year's worth of growth of secondary xylem produced by the vascular cambium. They appear as rings because the cells are smaller and therefore darker towards the end of the season when there is less water available. In the early season, the early wood of each growth year ring increment has larger diameter xylem cells, and therefore it looks like lighter wood. And that's why it resembles sort of rings in terms of the years of growth. I wanted to remind you that even really small plants can produce wood, and even though it's a small amount of wood, you can still see these yearly growth rings. That was a lot of information about the development of roots. Are there any questions? Yeah, I was wondering what kind of plants go through secondary growth? Yes, that is a very good question. Not all plants go through secondary growth. Remember how last week when we were or 2 weeks ago, when we were talking about cell tissues, I introduced this concept of eudicots and monocots. These are just two kind of major groups of plants that we talk about as botanists, and we'll be talking about throughout this semester. Well, monocots, things like grasses and lilies, these do not produce wood. Bamboo is technically not wood, even though you can build with it and it's very hard. It's not truly secondary xylem. Things that do produce true wood and true secondary xylem from a vascular cambium include the eudicots. When they do produce wood, they produce true wood, and things like gymnosperms, like conifers within the gymnosperms, which we'll be talking about throughout the semester. Any other questions? Yeah, I was spacing out for a while, and I was wondering, what is phyloderm, and where does that come from? No worries if you were spacing out. You can always go back through these videos, rewatch things, and pause them where necessary. But your question was a very good one. It was, where does phyloderm come from and what is it? So what we can ask on future kind of quiz questions, or maybe you'll be thinking about this in the future for some other reason, like the final, we may want you to sort of trace back individual tissues or cells. And so if someone were to ask, where does phyloderm come from? Well, directly, it comes from the cork cambium. Remember, it is the layer of cells that are dividing to the inside of the cork cambium. But then if we want to trace it all the way back, where does it come from in a root? Well, it comes from the cork cambium. Where does the cork cambium come from? It comes from pericycle. Where does the pericycle come from? Well, it's part of the vascular cylinder of a root, and the pericycle is derived from procambium. Remember that from the beginning, and the primary tissues and the primary meristems? Well, then where does procambium come from? That comes from the root apical meristem, all of the root apical meristems. And so you can trace back tissues and cells just kind of like what I did, but with all of the tissues we've talked about. I recommend doing that. Nice job learning about root development. See you at the next virtual lab.